Students, in this video now we will be talking about new drug development. So I have made a flow chart to explain you first and give you an introduction about what exactly we are going to discuss, what we are going to study. So for development of any new drug, these are the steps. First of all, there is synthesis of the compound. Now this synthesis of the compound can be from the plant sources or from the animal sources, or it can be chemically synthesized in the lab. It can be developed by molecular modeling, or it can also be developed by biotechnology. So these are various ways by which we can synthesize a new compound. After that, the preclinical studies are done, which are primarily animal studies, and in this we have in vivo studies as well as in vitro studies, right? So in these studies we do the screening, we do the evaluation, we test on our animal models. By screening we actually find out the effect, the pharmacodynamic of the drug, pharmacokinetics, toxicity testings, acute toxicity, subacute toxicity, short-term toxicity, chronic toxicity. These are the things which we actually study. Next, once the preclinical studies are done, then we actually give the drug for permission for clinical trials. So that is called as investigational new drug. So regulatory authorities are approached that yes, this compound has passed the preclinical studies, so kindly grant the permission for clinical trials. Now once the permission is granted, then the drug is formulated, proper dosage form is formulated, and then the clinical trials are conducted in a very phased manner, in a very logical, ethical, scientific phased manner. So the first step is phase 1 study. Phase 1 study is also called as human pharmacology and safety. This is also called as phase 1 is also called as human pharmacology and safety study, right? Then after that we'll discuss in detail. Then phase 2 study is called as therapeutic exploration and dose ranging. This is the phase 2 study. After phase 2 study we do the phase 3, which is called as therapeutic confirmation and comparison, right? Now when the drug passes through phase 1, phase 2, phase 3 and it passes all the parameters, then it is submitted to the regulatory authorities for as a new drug application. NDA stands for new drug application, for review and for grant of permission to be launched into the market. Now once the permission is given, then comes the phase 4, which is called as post marketing surveillance, or also called as data gathering study. So here basically the drug is marketed in the general public, and then side effects, idiosyncrasies, any unsuspected drug interactions then are finally detected at this stage. Then so we'll come to that in detail, and further decisions, then either it continues if there are no such side effects or adverse effect, or the drug is banned if there are intolerable side effects. So let's see all the things now one by one. So now the students will be like, why do we need to know about it? So we need to know about, we need to study this new drug development because the students or the future doctors should know about how the drugs are made, should know the process of drug making. So here what we are telling you is how a drug finally comes into the market, right? So the first step was synthesis of a new compound. Now the synthesis of new compounds is usually can be done through the plants. There are various plants, Ayurvedic plants we know that have got medicinal value. So a lot of plant sources are also there for the existing drugs. Then we can do a chemical synthesis also in the lab. So once suppose you identify a plant, then after that you take it to the lab and you isolate the chemical compound which is having the medicinal property, and that compound is called as the lead compound, right? And this lead compound is then finally used in the making of drug. So that is one way of doing it. Another way is that we can do molecular modeling. That means that we have got a lot of advances in protein chemistry, and computer aided elucidation can be used, has permitted designing of targeted compounds. So here we can make new compounds from the old one by doing some molecular modeling, like insulin. You see, when you will study, then insulin, the original insulin is modeled into various other types of insulins. right? And then the last way that it can be done is by biotechnology, by using the rDNA technique, that is the recombinant DNA technique. So a lot of drugs are made, like we know human insulin is made, human growth hormone is made by recombinant. A lot of monoclonal antibodies are now being made by recombinant technology. Then comes, after a new compound, a lead compound is identified, then we come to preclinical studies. Now in preclinical studies, these studies are basically studies which are done on animals. Okay, so these are the studies which are done on animals, and it is tested to expose their pharmacological profile complete, right? Now these animals which are used, which are the animals we can use? The animals are the small animals. So the animals that we can use are rabbit, mice, rat, guinea pig, and hamsters. Okay, so these are the animals that we can use. Large animals are now banned by the human animal rights activists, so we cannot use cat, monkey, and dog. Now what do we do in animals? What do? We first of all do the screening tests are done. There are various screening tests which are standardized, and they are used to basically find out the pharmacodynamics of the drug on the animal. That is, what is the action? Pharmacodynamics means action. Then we create models of diseases. In animals. For example, we want to test a drug. So various models of diseases are created, like for suppose we want to test a drug on lipids. So then we make animal, we increase the cholesterol by injecting fat diet in the animal and make it dyslipidemic, and then we test the drug of lipids on these animals. Then the third thing that we do is we also test the pharmacokinetic. How do we do that? We give the drug, inject the drug, and then we take samples at regular intervals, and these samples are tested for the concentration, for the dose at which the toxicity has developed. And then we have the toxicity studies. So in toxicity studies, we do acute toxicity study, in which a drug is given, escalating doses are given to see LD50 is identified here, that is dose at which 50% animals die. So this is the lethal dose. Then we also do subacute toxicity. A subacute toxicity study is done. Here ED50 and LD50 are identified. ED50 is efficacy, the dose at which 50% effect is found in 50% animals, and then the dose is calculated from here. Then we also do chronic toxicity studies. So the drug is given for 6 to 12 months, and we find out the toxicities if there are any on long-term use. Then here we also study for teratogenicity. So pregnant mice and pregnant animals are taken. They are first either pregnant, then mutagenicity and teratogenicity studies are done. Then again for long-term they are given, and carcinogenicity testing is done, that if they are used for long-term, then is there any development of tumor. So all these studies, all these tests on the animals, they are done as per the GLP, that is good laboratory practices. These are the guidelines, GLP, which is done for the animal experiments, especially the toxicity testings, right? Then next we come to the clinical studies. Now clinical studies, now there are 2-3 important things. They are done as per the GCP guidelines, that is good clinical practices. All the clinical trials are done as per the NEET rules 2019, that is new drug clinical trial rules 2019. So all the clinical trials are done as per these guidelines, plus they have to be registered with the clinical trial registry of India. So once they are registered, then only they are considered as valid. Otherwise you can do whatever you want to do, but they will not be registered trials. Now after the drug is tested in animals, we discussed as per the first here, that after the drug is tested in animals, then it is actually submitted to the regulatory authorities for permission, for grant of permission for clinical trial as investigational new drug. Once the drug is permitted, then a pharmaceutical dosage form is formed. कि देंगे कैसे, ऐसे powder form में तो नहीं दे सकते ना. So a dose is formed, a dosage is formed. Like it's a tablet, it's a capsule, it's a syrup, it's a inhaler, whatever is the dosage form, that is done. And then we start with the clinical studies, that is phase 1. So phase 1 clinical study is basically called as human pharmacology and safety study. Now this trial, this phase is basically done in human subjects or volunteers, right? Now when it is done in the volunteers, then they are given with the lowest dose, which is generally 100 or 1/10 of the highest dose in animals, which produced no toxicity. So a drug which produced a dose at which no toxicity, the highest dose at which no toxicity was seen, is taken, and uska bhi 100th या 1/10th is started, and then stepwise the dose is increased, dose is escalated to see the effect, and then it is seen that what is the drug, what is the dose at which ED50 is attained. ED50 is again when the 50% of the people they develop the effect. Then again, as the name suggests, it is a safety study, so adverse effects are also studied, that what is the maximum tolerable dose here and what is the minimal lethal dose. So that is also seen, that at what dose are we getting a side effect. And then all the vital parameters are basically measured, like blood pressure, respiratory rate, pulse, kidney functions, liver functions, all these are monitored, and it is seen that is there any change in the parameters. So this basically here we are studying the pharmacokinetics of the drug. Here no blinding is done. We don't blind the patient or the doctor. It is an open label study, right? So this is phase 1. Now we come to phase 2. So phase 2 studies are basically called as therapeutic, confirmation, and dose ranging. So phase 2 studies are dose ranging and therapeutic exploration. Now in this study we take patients, but patients are not much. There are only 100 to 500 patients, and it is usually a single center study, a small level study. And in this, the patients suffering from the target disease are selected, and then they are given the drug at a particular dose, which is considered to be effective dose, right, in a very controlled setting. Controlled setting में दिया जाता है. And again pharmacokinetics are studied, tolerability, that is safety, is studied, and pharmacodynamic, the effects parameters are studied. These trials can be randomized, these can be blinded, and these can be controlled. That means a placebo group can be added to see the effect of the drug. So this is a phase 2 study. After that we come to the phase 3. So coming to phase 3, now in phase 3 it is called as therapeutic confirmation study. So now this is a randomized double-blind controlled trial which is done on large patient base, larger patient group, which is around 500 to 3000, and it is a multicentric study. So it is done at various centers. So this phase 3 is basically done on a wide scale, and once the drug is found to be efficacious in phase 3, then application is given to regulatory authorities, which is called as a new drug application. And once it is approved, then then the package insert is made, how it is going to be marketed, which size of the tablet, the color, everything. Then package insert is made, and which includes the details of the drug, precautions, contraindications, storage conditions, and then it is submitted to the licensing authority. Okay. So with this, now when the permission is given, then it is launched into the market. मार्केटेड हो गई. अब मार्केटिंग होने के बाद देन कम्स दी phase 4. So phase 4, as we know, it is called as post marketing surveillance. Now in this, a group of physicians is identified, and through them on a structured proforma about the efficacy, acceptability and adverse effect in the real field situation. So this is more like a prescription event monitoring, right? Or you can also do a drug utilization study here, so that you are able to identify the side effects. Now what is the main purpose? Any long term side effects, any idiosyncrasies which we could not identify in the phase 3 trials, any unsuspected drug interaction, right? So these are the things which are normally studied in the phase 4 or the post marketing surveillance. Then further therapeutic in the trials, जो excluded group होता है, excluded group like pregnant and lactating mothers होती हैं, या children होते हैं, या patients with renal and hepatic diseases होते हैं. So in trials we could not test the drug on these patients, so these patients are then studied in phase 4. So in phase 4 we are able to study the effect of drugs on the excluded group also, and we come to know about their additional effects. So this was about the new drug development.